IPEX stands for immune dysregulation, polyendocrinopathy, enteropathy, and is an X-linked syndrome that is caused by mutations in the FOXP3 gene, which is a critical transcription factor required for Tregs. Patients often present with the triad of symptoms: autoimmune diabetes, enteropathy, as well as dermatitis. And it is a pretty severe disease that can be fatal if untreated. As a monogenic immune disease with limited treatment options, IPEX is an ideal candidate for a gene therapy approach. And at the bottom left, you can see our approach is to obtain patient HSPCs, hematopoietic stem and progenitor cells, gene edit them with a corrected copy of FOXP3, and then through an autologous transplant, return these cells back to the patient where they can engraft and produce healthy hematopoietic subsets. Our strategy involves CRISPR-Cas9, where we have a guide that cuts the endogenous locus. AAV6 is used to deliver the donor template that contains the full-length FOXP3, as well as an NGFR gene tag. You can see in the middle panel that once cells have been successfully edited, they start to express NGFR. NGFR positive cells can then be enriched for further experimentation. We found that we're able to use this strategy to edit different cell types, including Tregs, the Treg-like MT2 cell line, T effector cells, as well as HSPCs. In panels C and D, we look at an example of a patient 24 with an exon 1 skipping mutation causing their FOXP3 isoforms to run faster compared to the wild type. And once these cells were gene edited, you see that the full-length expression has been restored to this patient's cells. We also gene edited healthy donor T effector cells, and we found that FOXP3 expression over time, the cytokines that were produced, as well as the proliferation rates were similar across wild type mock, as well as gene edited T effector cells. We also looked at patient Tregs and found that after gene editing, patients' Treg cells had increased suppression capacity, which went from 4 to about 32%, which is pretty striking and within the normal range of a healthy donor's Tregs. And so this provides initial evidence that this might work in a patient setting. On the right panel, we looked at off-target sites and did next-generation sequencing and found that in all the predicted off-target sites, the percentage of off-target were pretty low. We then took this strategy forward and gene-edited HSPCs. For the in vitro aspect of this, we plated gene-edited HSPCs on methylcellulose media, and after 2 weeks, we enumerated the number of colonies and types of colonies that was formed and found no statistically significant difference between the wild type, mock, as well as FOXP3 gene-edited HSPCs. Confident of this strategy, we then moved on to engraftment models. We took day 3, day 4 irradiated NSG-SGM3 mice, engrafted them with gene-edited HSPCs, and looked at the engraftment over time. We found that gene-edited HSPCs were capable of multilineage engraftment, producing key hematopoietic subsets, including T effector CD4s, as well as CD25 FOXP3 positive putative Tregs. We found that the T effector cells were able to proliferate upon stimulation, while the CD25 FOXP3 positive Tregs, putative Tregs that were derived from gene-edited HSPCs were able to suppress activated T responder cells. So this was quite positive, and we believe provides preclinical evidence that this would work and that edited HSPCs are capable of engraftment and differentiation. Again, this really brings us back to our original aim, which is to provide a cure for every patient with IPEX